Hello students, in this video we'll continue with the chapter 8 cell, the unit of life. In the previous section we have seen the structure of the prokaryotic cell. Here I'll be discussing about the eukaryotic cells. Eukaryotic cells include all the protists, plants, animals and fungi. In eukaryotic cells, the cellular organelles are membrane bound, which is a clear cut difference between the prokaryotic and the eukaryotic cells. Membrane bound cellular organelles are found in the eukaryotic cells. And for example, the nucleus, which is having the nuclear membrane, then other cellular organelles like mitochondria, chloroplasts, lysosomes, ribosomes, endoplasmic reticulum, all these are found in the eukaryotic cells. So the eukaryotic cells are having much complex structure than the prokaryotic ones. And the eukaryotic cells are not identical also. Plant cells differ from the animal cells in having a cell wall which is made of cellulose, and plastids, chloroplasts are found in the plants, and large central vacuole which are absent in the animal cells. And in animal cells they have centrioles which are absent in the plant cells. In this slide, you can see the animal cell and the plant cell. You can compare the structures of these two cells, what are present and what are not there in the animal cell and the plant cell. Here you can have the comparison of plant cell and animal cell. First, we will study about the cell membrane. The detailed structure of the cell membrane was studied only after the invention of the electron microscope. But chemical studies on the cell membrane were done earlier, and the studies showed that the cell membrane is composed of lipids, and these lipids are arranged in a bilayer within the membrane with the polar head towards the outer sides and the hydrophobic tails which are arranged towards the inner part. So later, biochemical investigations revealed that the cell membranes possess proteins and carbohydrates as well. But the protein and lipids, their ratio varies considerably in different types of cells. Then, depending on the ease of extraction, membrane proteins can be classified as integral proteins or peripheral proteins. And these peripheral proteins lie on the surface of the membrane, while the integral proteins are partially or totally buried in the membrane. Here you can see the fluid mosaic model of the plasma membrane that was proposed by Singer and Nicholson in 1972. This is the most widely accepted model of the cell membrane. So, according to this fluid mosaic model of cell membrane, the quasi-fluid nature of the lipid enables the movement of proteins within the lipid bilayer. So this gives the fluidity to the cell membrane, and the fluid nature of a membrane is important for functions like growth, formation of intercellular junctions, secretion, then cell division, and such activities. Now, one of the most important functions of the plasma membrane is the transport of molecules across it. So, the plasma membrane is selectively permeable. It allows the movement of only certain molecules across it. Many of the molecules can move across the membrane without any expenditure of energy, without any requirement of energy. So, such a transport of molecules are called as the passive transport. And then there are other molecules which are required to transport from their lower concentration to higher or to lower concentrations also. So, in such cases, energy is required for the transport of such molecules. So, which is called as the active transport. Sodium ions, potassium ions are transported across the sodium potassium pump. So, that is done with the expenditure of energy. So, it is called as the active transport. Now, this passive transport, it is due to simple processes of diffusion or osmosis. But movement of water is by the process of osmosis. And the polar molecules cannot pass through the non-polar lipid layer. So then the active transport and the requirement of energy is there. In case of fungi and plants, there is a non-living rigid structure which is called the cell wall, which is forming as an outer covering for the plasma membrane. And the cell wall not only gives shape to the cell and it protects the cell from mechanical damage and infections. It helps in cell-to-cell interaction and provides a barrier to undesirable macromolecules. Now, the cell walls of young plant cell is the primary wall is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is formed on the inner side of the primary cell wall. And there is a middle lamella, which is a layer mainly made of calcium peptide, and which is holding the two neighboring cells together. The cell wall and the middle lamella may be having plasmodesmata, which connect the cytoplasm of the neighboring cells. Now let us study about endoplasmic reticulum. This is a network or a reticulum of tubular structures present scattered in the cytoplasm, and this endoplasmic reticulum divides the intracellular space into two distinct compartments. That is one compartment which is inside the endoplasmic reticulum and which is outside, that is in the cytoplasm. So two compartments are there. And this endoplasmic reticulum provides a skeletal framework to the cell. Materials move around in the cell through this endoplasmic reticulum. Now, there are two types of endoplasmic reticulum depending on the presence or absence of the ribosomes. Now, those endoplasmic reticulum where the ribosomes are attached on the outer surface, those endoplasmic reticulum are called as rough endoplasmic reticulum, and this rough endoplasmic reticulum are involved in the protein synthesis and secretion. Now the second type is smooth endoplasmic reticulum. So there is no ribosome attached on the outer surface of the endoplasmic reticulum, so they give a smooth appearance. So they are called smooth endoplasmic reticulum, and they are the sites of synthesis of lipids. Here you can see the differences between smooth and rough endoplasmic reticulum. And you can see the diagram of the endoplasmic reticulum, where you can see this network of tubules are extensions of the nuclear envelope. The next endomembrane system is the Golgi apparatus or the Golgi complex, which were observed by Camillo Golgi. And these are sac-like structures which are stacked one above the other. Uh, they consist of many flat disc-shaped sacs or the cisternae of 0.5 micrometers to 1 micrometer in diameter, and they are parallelly stacked to each other, and variety or varied number of cisternae are present in a Golgi complex. And these Golgi cisternae are concentrically arranged near the nucleus with convex sides which are called as cis or the forming phase, and the concave side which is called as the trans or the maturing phase of the Golgi complex. These are entirely different, and but they are interconnected. The Golgi complex performs the function of packaging of materials which are to be delivered either to the cell itself, or it has to be sent to some other place also. Materials to be packaged in the form of vesicles from the endoplasmic reticulum, and they fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. So, a number of proteins synthesized by the ribosomes of the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus, and then they are released from the trans phase. So, this Golgi apparatus or Golgi complex is the site of formation of glycoproteins and glycolipids. This is the diagram of the Golgi apparatus, where we can see the cisternae and the vesicles, the concave side which is the cis side, and the other opposite side is the trans phase where the vesicles are formed. The secretory vesicles are formed from the trans side. Lysosomes are another kind of membrane-bound vesicular structures which are formed by the process of packaging in the Golgi apparatus. So the isolated lysosomal vesicles, they have been seen to be very rich in hydrolytic enzymes like hydrolases, lipases, proteases, and other hydrolyzing enzymes. They are optimally active in acidic pH, and these enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids also. So these are responsible for the degradation of worn out cells. So lysosomes are called as the suicidal bags of the cell. So their main function is intracellular digestion, and this intracellular digestion is by the phagocytosis or the pinocytosis. Vacuoles are the membrane-bound space found in the cytoplasm, containing water, sap, various kinds of excretory products, and other materials which are not useful for the cell. The vacuole is lined by the single membrane which is called the tonoplast. In plant cells, the vacuoles are very large and they occupy up to 90% of the volume of the cell. In plants, the tonoplast helps in the transport of various kinds of materials against concentration gradients into the vacuole. So, concentration is higher in the vacuole than in the cytoplasm. And in amoeba, so unicellular organism, the contractile vacuole is helping in the excretion. And in many protists, the cells, food vacuoles are formed by engulfing the food particles. So, contractile vacuoles, food vacuoles. So, many types of vacuoles are there which are differing. Mitochondria is one of the self-replicating organelles of the cell, and these are normally visible under the microscope. The number varies in the cells depending on the physiological activity of the cell. Shape and size of the mitochondria also vary a lot. Typically, the mitochondria are sausage shaped or cylindrical in shape, having a diameter of zero point 0.2 to 1 micrometers, that is average. Length is 1 to 1.04, like up to 4.1 micrometers. And these are double membrane bound structures, and the outer membrane and the inner membrane. Between the outer and the inner membrane, there is a lumen, which is making the mitochondria into two compartments, the outer compartment and the inner compartment. Now, the inner compartment is called as the matrix, and the outer membrane forms the boundary of the organelle. Now, the inner membrane having infoldings into the matrix, which are called as the cristae. Cristae are the infoldings of the inner membrane into the matrix. So these cristae are there to increase the surface area. And the mitochondria is responsible for cellular respiration and produces the energy for different activities, and the energy produced is stored in the form of ATP. So mitochondria is called as the powerhouse of the cell. Now, the two membranes, the outer and the inner membranes, they have their own specific enzymes which are associated with the functioning of the mitochondria. Now, the matrix possesses DNA molecules. Sometimes you will find a few RNA molecules also, and the matrix contain ribosomes also along with the DNA and the RNA. So, sufficient proteins can be synthesized within the mitochondria itself. So, this mitochondria is called as self-replicating organelle because it has got circular DNA, RNA, as well as ribosomes for protein synthesis, so it can replicate on its own. And the mitochondria divide by fission during the cell division also. Here in the diagram you can see clearly the outer membrane, the inner membrane, and the matrix, and the infoldings or the cristae of the inner membrane. The plastids are also double membranous structures which can also self-replicate. These plastids are found in the plant cells and in euglenoids also. These plastids contain specific pigments in them, so they provide the pigments, give them colors. So based on the type of pigments present, the plastids are classified into chloroplasts, chromoplasts, and leucoplasts. The chloroplasts, they contain the green coloring pigment chlorophyll and carotenoid pigments, and these pigments are responsible for the photosynthesis. Now, the second kind, that is the chromoplast, they contain fat soluble carotenoid pigments like the carotenes and the xanthophylls, and This provide the plant yellow, orange, or red color. Then the last group, that is the leucoplasts. These are the colorless plastids, and they are storing some nutrients like starch or oils and fats or the proteins. Now the leucoplasts, which contain or which store starch or carbohydrates, are called as amyloplasts. Then there is elaioplast, which store oils and fats, but the aleuroplast, they store proteins. This coming to the structure of plastids. Chloroplasts are lens-shaped, spherical or discoidal in shape, and the mitochondria are double-walled or double membrane-bound organelles, and the inner membrane is relatively less permeable. The inner membrane contains the space which is called as the stroma. And inside the stroma, a number of flattened sacs, which are called as the thylakoids, they are arranged in the stroma, arranged as stacks. So these stacks of thylakoids are called as the grana. Now there are flat membranous tubules which connect the thylakoids of different grana, which are called as the stroma lamella. So the stroma lamellae connect the thylakoids of different grana. And the stroma of the chloroplast contains various enzymes which are responsible for the photosynthesis. And the chloroplast contain small double-stranded circular DNA molecules as well as ribosomes. The chlorophyll molecules or chlorophyll pigment is present inside the thylakoids of the chloroplast. Next is the ribosomes. The ribosomes are the granular structures which are found as dense particles, and they are composed of RNA and proteins. These are not membrane-bound organelles. They are not surrounded by any membrane. The eukaryotic ribosomes are ATS. When it is in prokaryotic ribosomes, it is 7DS. S is the Svedberg unit, and the sedimentation coefficient it represents, and depending on the density and size, the ribosomes are given this unit. Both the 7DS and the 8DS ribosomes are formed or composed of two subunits. And these ribosomes are involved in the protein synthesis. Next is the locomotory organelles of the cell. They are the hair-like outgrowths of the cell membrane, cilia and flagella. They are hair-like outgrowths for locomotion and movement. The cilia are small structures which work like oars, causing the movement of the cell forwards or movement of the surrounding fluid in which the organism lives. Flagella are comparatively longer, and they are fewer in number also. The electron microscopic study of the cilium or the flagellum show that they are covered with a plasma membrane, and the core of it is called as the axoneme. which possess a number of microtubules running parallel to the long axis. So the axis is called as the axoneme, and the axoneme has 9 pairs of doublets which are radially arranged spherical microtubules and a pair of centrally placed microtubules. So such an arrangement of microtubules is called as 9+2 arrangement. The central tubules are connected by bridges and is enclosed by the central sheath, which is connected to one of the tubules, which peripheral doublets. The peripheral doublets are the radial spokes. They are connected to the central sheath. So there are 9 radial spokes, and the peripheral doublets are also interconnected by the linkers. Now the cilium and flagellum emerge from the centriole-like structure called the basal body. So these are emerging from the basal bodies. Now this is a paramecium where you can see the cilia on the surface of the cell. So, and this is a euglena where you can see the flagellum, which is much longer than the cilia. And the next organelle is the centrosome, which is normally seen in the animal cells only. And the centrosome contains 2 cylindrical structures which are called as the centrioles. The centrioles are surrounded by pericentriolar materials, and the centrioles in the centrosome lie perpendicular to each other, and they are made up of 9 evenly spaced peripheral fibrils of tubulin. The protein is called tubulin, and each of the peripheral fibril is a triplet. So in case of cilia and paramecium, it is a doublet, but here in centriole or centrosome, it is a triplet. And the adjacent triplets are also linked. And the central part of the proximal region of the centriole is proteinaceous, which is called as the hub, the central region of the centriole. Then the hub is connected with the tubules, the peripheral tubular triplets by radial spokes, and the centrioles form the basal body of cilia, the flagella. And the spindle fibers, which are formed during the cell division, is formed from the centrioles. Here you can see the diagram of the centriole. The peripheral triplets which are connected to the central hub through 9 spokes, and the triplet fibrils are also interconnected.